The broadcast is now starting. All attendees are in listen-only mode.
Marco, can you hear me?
We can all hear you.
Great. Okay, perfect. All right. Welcome, everyone. This is a redo of our first unsuccessful attempt at doing the webinar on the testing of honey using palynological techniques. My name is Juliana Rangel. I am associate professor of entomology, and I am the honeybee specialist here at A&M University. And I'm here with my student Pierre Lau, who will be talking to you about how honey is tested. So the collaborators on this project are, of course, the Texas Beekeepers Association, who received a grant from the Specialty Crops Program of the Texas Department of Agriculture. And we're also collaborating with Dr. Marco Palma from Agricultural Economics here at A&M, Dr. Deborah Delaney, who is in the Department of Entomology at the University of Delaware. This is the first of a two-part webinar series on this grant, increasing consumer awareness of the economic and health benefits of real Texas honey through a multi-stage research and marketing campaign across the state. So one of the objectives of the grant was to create educational tools and curricula to disseminate strategies, marketing strategies, and also honey testing strategies to Texas honey producers and the general public. So that's what we're doing right now with these webinars. Another one, of course, is to test honey using microscopic determination of pollen coefficients to determine the regional denomination of origin or an approximation of that for real Texas honeys. And that's the project that we are finishing up here at Texas A&M University. So we're determining the major plants in Texas honeybees that produce nectar, that honeybees produce nectar from, and to develop a standard for analyzing real Texas honey. Another of the several objectives of this webinar is to organize working groups with honey producers that will help assess the economic value of Texas honey production and identify which labeling and product attributes are most important for the Texas honey market using a consumer willingness to pay economic study that is led by Dr. Marco Palma. So that webinar is going to be on next Thursday, September 27th at 10 a.m. Central Time, and it's going to be pretty similar to what we're doing today, but it's going to be on marketing techniques for honey and the consumer willingness to pay information. And so we hope that you can join us next week. But if you are unable to join us today or next week, we will have links to the recordings that you can access later on at your leisure. So we will share those links with you later on after we finish the webinar. So without further ado, I want to thank you for being here and for supporting our research program and the Texas Beekeepers Association Real Texas Honey Program. And now I will leave you with Pierre Lau, who is the person that has been studying under the umbrella of Dr. Von Bryant, who is one of the world's leading expert on honey testing, on how honey is tested to determine the floral origins of nectar. Oops, I don't know how to go next.
All right, great. So I'm gonna go and get right into this presentation. Once again, our objective here is to help Texas beekeepers to market their honey to improve their sales, and we're going to do this by one of the ways we're going to do this is by doing research, and we're also going to help educate the public to improve public awareness of the honey market and industry. So I'm going to give a brief introduction just on what honey is and how it is defined. But this is taken straight from the European Union, but their standards are one of the highest in the world. And they define honey as the natural sweet substance produced by Apis mellifera bees from the nectar of plants or from the secretions of living parts of plants or excretions of plant-sucking insects on the living parts of plants, which the bees collect, transform by combining with specific substances of their own, deposit, dehydrate, store, and leave in honeycombs to ripen and mature. So I'm just going to break this down real quick, this entire quote. But the first part here, the thing that stood out to me is they specified Apis mellifera bees. So this is for honeybees specifically. Okay, so even though certain species of stingless bees will produce honey in pots, they're defining honey as honey produced by Apis mellifera bees. Okay, and these are going to be from nectar from plants or excretions from plant-sucking insects. So this will include honeydew. So honeydew honey from aphids, that's also included into the definition of honey. And the rest of this quote just talks about the entire process of how honey is made, the natural process. So a bee is going to collect it, secrete enzymes of their own, and deposit it, dehydrate it, and reduce moisture content, and let it ripen and mature. Okay. In addition, they also have another part that says that honey sold as such shall not have added to it any food ingredient, including food additives, nor shall any other additions be made other than honey. No pollen or constituent particular to honey may be removed except where it's unavoidable in the removal of foreign inorganic or organic matter. So all this is saying is honey should be at its purest form. You should not add any additional substances such as additives and high fructose corn syrup or sugar water to get an economic profit out of the honey. In addition, they also say that you should minimize removal of any particle in the honey, and what they're referring to here would be the pollen. So pollen is going to be naturally found in honey, and it's going to be a very important part of honey, as we'll talk about later in this lecture or in this webinar. Okay, this is just a table summarizing what honey is, and really it's about 70% sugar. 70% of that's going to be your two types of sugars, and then it has about 18% water, and then you have a bunch of other minor sugars and minerals, amino acids, proteins, and it has an average acid and pH value. So nutrient-wise, this is just like a sweetener, a carb, but honey, as we know, is a lot more than that. And one of the reasons is because their glycemic index is very low. So what that means is that it can, for people with diabetes, it will help them maintain a lower blood glucose level compared to sugar. Okay, so this is just a table showing some common honeys, and most of them actually have a lower glycemic index right here compared to sucrose and glucose. So honey is a great source as a sweetener for people with diabetes. But honey is also really well known for anti-blank activity. And yes, it's fill in the blank because you can fill it in with almost anything you want. It's antimicrobial, antifungal, it can and even be antiviral as well. It can inhibit the growth of microorganisms, fungi, and certain strains of viruses. And a lot of that's attributed to phenolics and flavonoids. These are secondary plant compounds that's antimicrobial, and these are the same compounds that you would find in a lot of some of the foods that we eat, such as our blueberries, for example. Honey also has a very low water activity. We mentioned that they cap it and dehydrate, and that's to get the moisture content low. So with this low water activity, it can help inhibit microbial growth. It can help produce glucose oxidase, which will produce hydrogen peroxide, and hydrogen peroxide is an antiseptic. So we use that to clean. We know that we use that to clean our wounds, so bees use that as well in the colony to keep things sterile. And it has antioxidant functions, which can help prevent cells from damage caused by oxidative stress. This table over here is just a list of bacteria that were found to be sensitive to and inhibited when they're exposed to honey, and this is just a great list that's applicable to a lot of us. Oops. As a result, honey has been exploding in popularity. The media covers it all the time. When you just look up honey on Google search, you have a lot of applications of honey, whether or not some are completely true and well enough researched to be factual or not. There is a lot of coverage on honey, and there's this explosion right now of how honey can be used to benefit our health. So it's just driving up the demand for honey exponentially. So this brings us to our honey industry. So we have about 2.5 million colonies divided among commercial beekeepers, part-time beekeepers, or sideliners, or hobbyist beekeepers. So these are defined. So commercial beekeepers are the ones that are going to be trucking colonies from state to state, pollinating different regions and different types of crops in different regions. Sideliner beekeepers are also pretty large in scale, but they are not as big. And hobbyist beekeepers, you can think of that as like your backyard beekeepers. In terms of population and numbers, most beekeepers are actually going to be hobbyists. That's because of how big beekeeping is nowadays. However, the majority of colonies are going to be managed and by the commercial beekeepers. As a result, the commercial beekeepers still produce about 60% of the honey in the United States, and about half of the honey produced in the US we packaged in large corporations such as Sue Bee and Clover Maid. To make matters worse with their rising demand for honey, there's also a decline in honey production in the US over the last 24 years. You've heard since like 2006, 2007 that bees have been in decline. So our bee populations haven't been rising steadily over the past dozen years or so. In 2017, the US produced about 147 million pounds of honey, which is actually 9% less than the year prior to that. And a lot of decline can be attributed to declining honeybee health. This is just a graph I found from Ward 2014, showing the demand for honey. On the x-axis, you just have time, and then the y-axis, you have an index that looks at the growth in demand since, and this is goes back to 2008. There are spikes and it goes up and down, but overall, you see the general trend. Demand for honey has been increasing over the last 10 years or so. On this graph, on your x-axis, you have time once again, and on your y-axis, you have honey produced. And the lighter line is going to be, the light gray line is going to be US honey production, and the dark gray line is going to be amount of honey imported. And you can see in this graph that over time, since all the way back in 1986, the amount of honey that's being produced in the United States has dropped gradually all the way until 2012, whereas in order to meet that demand, we had to increase our imports of honey from other countries to meet the rising consumer demand. And a lot of that, you can attribute that to the time period when Varroa first got introduced. Honeybee colonies started struggling during that time, and even now today. And that's like the #1 culprit of bee colony losses. So this trend is not surprising. So in terms of imports in 2012, the United States imported almost 300 million pounds of honey, which represents close to 70% of the total consumption. And a lot of the major honey exporters that import into the US is Argentina, Vietnam, India, Brazil, and Canada. So this bullet point is missing one major country that it's not included here, and that's China. And that's because Chinese honey in the early 2000s started becoming heavily taxed because of dumping. China produces millions and millions and millions of pounds of honey, and they have a huge industry because of all the canola and agriculture that they have. So they have a surplus of honey that they actually have to get rid of. So they sell it in very, very low amounts, and they sell the honey very cheaply, and it would outcompete U.S. honey. However, unfortunately, even though that it's heavily taxed, the Chinese honey is still finding its way into the United States. Here, this was an article that was just published this year, stating exactly that. The Chinese have found a way to evade this import tariff, and they do this by sending out their honey to a third-party country such as India, and they get to relabel the honey and everything, and then they sell it back to the United States as Indian honey or Argentinian honey, and so on and so forth. So they avoid this heavy tax because of that, by doing that. So this is the national standards from China for honey. And it's quote, a natural sweet substance produced through fully brewing when the nectar, secretion, and sweet deposits from plants are gathered, mixed with secretions of their own, modified and stored in the honeycomb by honeybees. Overall, it's okay. I mean, it describes the process of collecting nectar from plants and storing it. But one thing that this does not have, that's omitted, that the European Union has, is that it does not specify that you can add any additives or other types of sugar into your honey and dilute it. That's something that they don't have. So you can add other sources of sugar or sweeteners to dilute the honey, and you can still technically call it honey. And this is very important because Chinese honey is actually discovered to be adulterated and contain impurities such as antibiotics and heavy metals. So this, in addition to the fact that it's they produce at such large amounts. This is another issue that we have with Chinese honey. This is the brassica field or canola field that in China, and it's literally like a field of gold. So they produce a lot of canola honey, and canola honey is very unfavorable because of how the sugar spectrum is. The honey will crystallize very fast, which makes it undesirable for consumers. So once again, it started getting heavily taxed in the beginning, in the early 2000s, and they got around that by shipping it to other countries. And every year, the Immigration and Customs Enforcement Department in Houston, they seize millions and millions of pounds of illegal Chinese honey, but we are positive that there are a lot more coming in than we can catch. Another important concern that we have about Chinese honey is chloramphenicol. This is an antibiotic used to treat typhoid, systemic salmonella, meningitis, and plague. And it's also a carcinogenic to us. It can cause leukemia, and it can suppress the bone marrow. I mean, this obviously sounds like a human-related antibiotic drug. Why are we seeing this in honey? In the late 1900s, beekeepers in China had a really bad case of American foul brood, which is a very deadly disease for honeybee colonies. When a colony gets it, the method of treatment is you're supposed to burn the colony and destroy it and get rid of the colony. So there were very little restrictions in China on how people can, how beekeepers can treat their colonies, and they found out that this antibiotic works, so they started using this. And to this day, even I'm not sure what the status of this is, but to this day, this is still present in all Chinese products. You find it in Chinese honey. You find it in royal jelly and other bee products such as wax. So obviously there's a lot of concern here for imported honey, especially from China. But unfortunately, there is no truth in labeling in the United States. So this is what you'd see at a typical grocery store. You see honeys that have these buzzwords to increase their value. If you label something with local, unfiltered and raw, you can put the USDA organic sticker on it, or non-GMO. You can raise the prices of your honey pretty significantly, because that's what people are looking for. Unfortunately, as I mentioned, there is no truth in labeling there. So our objective here, I'm just going to reiterate this, is to scientifically identify how to improve marketing for real Texas honey, which can help improve honey sales and honeybee education to the public sector. And for this first part of this webinar series, we're going to talk about how the research that we're doing involving identifying the major plants and the types of honey that you would normally find in Texas, and also developing a standard for a chemical spectrum that we can use in the future. So what we did over the past year is we we wanted to collect samples of honey from all over the state of Texas. So we have multiple different, I think 10 different ecoregions in Texas, and we want to collect honey samples from all over Texas and analyze it then. And that's because each region can have different types of plants, have different environments. So bees in each particular region will be producing different types of honey. Okay, so we sent out participant packages to beekeepers all over the state, and they received 2 sample baggies to put a sample of honey in, and then they put information about their location, when it was extracted, etc. And they shipped it back over to me. And there are a few ways that we can approach identifying honey. Okay, the first one is organoleptic. This is using our senses to identify the honey. Okay, so that's to look at the color of the honey, maybe the smell and aroma might give you an indication of what it is. But obviously for honey, it's not very reliable. I'm going to focus on numbers 2 and 3. And these are your physical and chemical characteristics, and also microscopy and paleontology in this for the rest of the webinar. So I'm just going to gloss over that real quick. But the last option would be the fourth option that I have here is DNA metabarcoding. That involves amplifying a loci, universal loci on of the mitochondria, and then amplifying that and then obtaining a list of the types of plants that were found that honeybees are collecting from. So first I'll talk a little bit about medicinal paleontology. This is the study of pollen contained in honey to better understand honeybee foraging ecology. Okay, so that image is just an SEM picture with a little bit coloring of different types of pollen grains. And we can do that because the taxa of pollen that honeybees collect that you see will indicate the type of floral resources that the bees are using. So you're kind of like playing forensics here. You're tracing everything back to where it was coming from. And we can do this because each individual species of plant will produce a unique pollen grain to their species. So that's why we can trace it back to the particular plant. And we mentioned earlier, there's a lot of important consumer value because we have different types of honey. So clover honey would be your very common store-bought honey. You can find this for around $10, like for a pound. Then you have sourwood honey from North Carolina. This is considered to be one of the tastiest types of honey from beekeepers. And this is obviously going to go for a little bit more than that. And then we have Manuka honey, which is from New Zealand. That's highly regarded because of its antimicrobial activities, and it's supposed to be way better with the antimicrobial activities compared to other types of traditional honey. And most leading honey producers, especially in Europe, actually require accurate labeling of the honey, but the United States does not. So how we do this is we first acetylize the pollen grains. So this is a 901 solution of acetic anhydride and sulfuric acid. So it's a very strong acid that's used to break down the pollen kit of the pollen grains. So the pollen kit is essentially the armor of a pollen grain. So for plants to reproduce, they use pollen. They have to move it from one plant to another, one flower to another flower. And during that time, they're going to be exposed to the environment. So they have this pollen kit or this armor to help protect the pollen's cytoplasm from the environment and from degrading out there. And so we use this very strong acid to remove that. And that's because we use the morphological characteristics underneath to identify what pollen it is. So here is an example of rose pollen that's unacetylized and unstained. And even with years of experience, and even if you ask Dr. Brian, we might be able to guess what family this is, but we have no confidence in identifying what that is unless you collect it from the plant directly and you know that it's where it's from. Okay. On the other hand, this is an image of an acetylized pollen that's actually slightly overstained with safranin, but you still see the characteristics that we use for identification really well. You see the apertures. If you rotate a little bit, you see like a pore over here. So this one will have 3 apertures and 3 pores. You can see like the surface structure of the pollen grain. So this one's going to be like kind of wavy. So that's called striate surface structure. And both of these are actually going to be in the same family. So how this works is we first take 10 grams of honey and we diluted it a little bit with warm water. And we added a lycopodium spore, which I'll talk a little bit about later, and added ethanol into the honey. After a series of centrifuging and extracting the pollen from the honey, we add some glacial acetic acid. This is to remove the water content from the honey. And we want to do this because the acetylysis mixture I was talking about, it reacts very violently with water. So for our own protection, we use glacial acetic acid first to remove that so that the samples do not explode in our faces. So after we do that, we make the acetylysis mixture and we pour it in, and then we let it react on a heating block for about 10 minutes at 80 degrees Celsius. After washing and after a series of washing the samples off, we stain it, and then we mount them on slides to look at under the microscope. So to analyze our samples, we count 200. We did a 200 pollen grain count per 10 grams of honey, and this was established from one of Dr. Bryan's previous studies. That's because in order to capture all the data, all the taxa in honey, you'd actually have to do over 1,000 counts of pollen grains in order to capture everything. But as you go past 200 pollen grains, you really start getting marginal returns after that. So there's the trade-off for counting more taxa and spending more time on it the trade-off for encountering more taxa would be to spend more time, a significantly more amount of time. After that, we summarize our data by categorizing it based on pollen abundance from our 200 grain pollen count. And we base this off Louvois et al. 1978. So pollen types that were over 45% abundant or predominant in a sample. And whenever you have a predominant pollen in a sample, that's when the International Honey Commission will officially call it a monofloral honey. So if you want to call something clover honey, it had to have over 45% based on that criteria. Secondary pollen types will have 16 to 45% of that particular pollen in the sample. And the important minors are 3 to 16%, and minor pollen types are under 3% of the pollen in the sample. And once again, honey is considered unifloral by the IHC when you have a pollen type that's represented at over 45%. The next thing I want to talk about is pollen concentration. I mentioned how we spiked our honey samples with lycopodium. These are spores that are not used by bees at all. So it's used as a control for us to make sure nothing went wrong while we're processing the sample. So if I'm processing a honey sample and I don't see any pollen at all, and I don't see any lycopodium spores, that means something went wrong on my end while I was processing the sample. However, if I was processing the sample and then I see pollen, I see no pollen, but I see lycopodium spores, that tells me that the honey sample just had no pollen. Okay, so this is just to make sure that what we're doing is correct. Okay, these are your spores and the plant it comes from. And the second thing that we do after we, in addition to using it as a control, is we use it to calculate concentration values. So we can use this formula here, where the number of pollen grains counted times the number of lycopodium spores added divided by the number of lycopodium counted. That will give us the average, the amount of pollen in a 10-gram sample of honey. So this over here is just a chart from one of Dr. Bryan's study, and it just has, you have your county, your region, your grains, and the lycopodium spores that they counted, and also the number of lycopodium spores that they counted, and also the pollen concentration value that was calculated from that. And the pollen concentration value can actually be broken down to 5 different categories that I'm going to go over. The first category is category 1. Okay, this is your samples with less than 20,000 pollen grains per 10 grams of honey. And these include, samples that are category 1 include samples that may have been filtered, samples from floral sources that produce very little pollen, such as mint, samples that were being fed sugar water during the nectar flow, or samples that are just straight up mixed with sugar or high fructose corn syrup. Okay, so a lot of the filters that we use, even though the size of the filter itself is like 500 microns or much larger than what the size of a pollen grain is, which is around 20 to 100 microns. Any type of filtering, Dr. Bryan was looking at honey, and he mentioned that any type of filtering will remove most, if not all, of the pollen in a sample. And he wrote an article about that, and a lot of that can be because the wax pieces or bee parts will end up getting clogged, and the honey and the pollen will get caught along with that. Even if, this is a very extreme case of your filter getting clogged, but even when it's relatively free-flowing, the pollen will easily catch on to the sieve itself and get caught there. So that any type of filtering will actually affect the pollen concentration value of your honey. Then we have categories 2, 3, 4, and 5 honey types. So category 2 honey types would be in between 20,000 to 100,000 pollen grains. And most of the honeys that's produced in the world will actually fall into this category. And that would be like your clovers, some of your mustards are also going to be in this category. Category 3 pollen types, this is your loosestrife, your purple loosestrife, will be between 100,000 to 500,000 pollen grains. Categories 4 and 5 are going to be from plants that have very little pollen relative compared to the amount of nectar that they produce. And category 5 pollen types specifically, ones that have over 1 million pollen grains per 10 grams of honey. Those are going to be from plants with a tremendous amount of pollen relative to the honey, the nectar that they produce. It can also be from honey where there were pollen cells in the frame with the honey itself. So when beekeepers extract the honey and put in a centrifuge and you spin it out, the pollen in the cells can come out as well, and that can actually increase your pollen concentration in your honey sample and affect the true pollen concentration of what your honey was. So here's an example of what we would see when we're looking at it under the microscope. This picture over here will have a large pollen grain here. This is called from magnolia. As well as larger pollen types that we would see in our samples. This tiny pollen grain on the bottom is willow. This is a trichoporic pollen type, which means that it has 3 pores. It's also going to have 3 scars as well. This is crepe myrtle pollen. Oops. No. So that one is crepe myrtle pollen. And your last one in the corner is sweet clover pollen. So just a few other pictures of pollen to show how diverse they can look. Because once again, each individual plant will produce a unique pollen type. These are your rose. This larger one specifically is prunus. So you can think that's the same genus as your cherry trees. On the corner, you have oak. You have lilac on the upper left-hand side of this image. You have rose, which is sumac. You have daisies and weeds. And then the last one here, you have a sweet gum on the bottom corner. Just to illustrate the diversity on how some of these pollen types look. So a few years ago, Dr. Bryant actually did part of the objective of our study, but just a subset. He looked at East Texas honey, several counties in extreme detail over the course of 7 years. So he analyzed 37 honey samples from East Texas using both a light microscope and a scanning electron microscope. And he was able to identify taxa belonging to 61 families, 104 genera, 85 species, and each sample contained 17 to 52 taxa. And these are pretty high numbers because he got to a very high level of detail and taxonomic resolution when he was analyzing. So this entire study took him about 7 years to analyze it, and it took that long because he used a combination of a light microscope and SCM to look at pollen and get it down to the species level identification. Okay, so I'm just gonna go over his results real quick. This image, I'm gonna show a bunch of pictures of different plants, and those are gonna be in descending order on the most abundant pollen types that he found throughout the study. So here's the top 7 pollen types that he found in East Texas. He found Alabama supplejack, eastern poison ivy, black willow, grass, black gum, New Jersey tea, and white clover. And then he also found American buckwheat vine, Carolina buckthorn, crimson clover, oak, sunshine mimosa, carrot family, beautyberry, grapevines, ragweed, sedges, rattlepods, lizard's tail, holly, and tooth leaf. So a lot of this he got down to the genus or species level. He spent 7 years and got this to very high detail. Okay, so for our study, within the last year of processing and analyzing samples, we found 130 unique plant taxa so far, and 33% are predominant honey samples. Okay, so we had Prosopis, which is your mesquite, and then you had Bershemia, which is your Alabama supplejack. Diospyros was your persimmon. You have your daisies and weeds. Lagerstroemia is actually an interesting one. That's crepe myrtle. But the thing is, crepe myrtle and also next one, grass, does not produce a nectar. They produce only pollen. So probably the pollen from the cells got out and got into the honey somehow. And Prosopis mesquite, you have a buttercup family in Ranunculaceae. Tridaca, that is your Chinese tallow. That was one of the predominant types. And sweet elm. So this list over here were the honey types that were predominant. And then we had 46% of our samples that had under 20,000 pollen grains per 10 grams of honey, which places them in category 1. So if you remember, category 1 are the honey types that were either highly filtered, were fed sugar water during the nectar flow, were from plants that were producing not much pollen, or it can be from adulteration where they mixed it in with other sources of sweeteners. This is a graph of breaking down all the taxa that we found in descending order. So we have the relative abundance of the pollen type that was found throughout the entire state of Texas, and on the x-axis you have the taxa. Throughout the entire state, the top plant that we found was actually sweet elm. That was the top pollen that we found in honey. And then we found Prosopis, your mesquite, and then daisies and weeds. Number 4 is going to be your Chinese tallow. That was a very important pollen type for certain regions. Number 5, we found crepe myrtle. 6 is grass. 7's sumac. 9 is going to be your clovers. 8 is your clovers. 9 is your willow. 10 is your oak. 11 is your Alabama supplejack. And 12 is going to be in your rose family. Okay, so the information that we have is really cool right now, and these are just preliminary results of about 70 or so of our samples. We have a lot more coming in that's already analyzed, but there are a lot of limitations to using this method for analyzing honey. Okay, so there are a lot of different methods of actually processing honey. Exotolysis is going to be one of your better ones if you want to look at the pollen grains in detail. But if you're not using that, there are other methods floating around that may not give you the same resolution for accurate identification or confidence in identifying them. And the pollen concentration values can have multiple interpretations with that. So we saw almost half of our samples were category 1 pollen honey types. And that can mean a lot of things. We can't say that honey was adulterated because maybe the bees were just whoever bees those were just foraging on sugar water throughout that time. Maybe they were scavenging in trash cans. Or maybe it was the honey sample was filtered to an extent. Any of those could have attributed to our high number of category 1 honey types. In addition, medicinal palynology is a long process, and it requires a very high level of expertise to identify the pollen types. And this information can definitely be supplemented with SEM or DNA metabarcoding to obtain taxonomic resolution. I briefly touched upon both of them, but SEM will be using your scanning electron microscope to look at the pollen in more detail, so you can go further into the economist key to more accurately analyze it with more confidence. And DNA metabarcoding is actually really neat to get a lot of species level identification at the lower taxonomic resolution. However, at this point in time, DNA metabarcoding does not produce any quantitative results. So a lot of times people suggest to combine this with medicinal palynology. And it also will produce quite a few false positives, so it'll misidentify or come up with a species of plant that you know for sure is not there. There's still some details that need to be worked out with DNA metabarcoding to use in the future. The next part I'm going to talk about now is the chemical analysis part of the study. And there are going to be targeted approaches and non-targeted approaches, but the common targeted approaches is look at water content analysis. So if your honey, how much water content's in your honey? Usually it should be around 17%. If it's over 20%, that can be an issue because there's too much moisture content, and you can get that by when beekeepers pull their honey and harvest it before it's fully ripe. And it's beneficial to an economically driven beekeeper because you're going to have more, you're going to get more bang for a buck in terms of honey. You're going to have more moisture in it, and you get more of it. It's not, it's going to be more diluted. And then the next one's ion exchange resin analysis. This is to target certain types of resins. So remember I talked about the certain drugs that we found, antibiotics found in honey. On Alibaba.com, if you look at it, if you search it up, it says Alibaba is like the Amazon of China. But if you look at it, they sell this resin that is marketed to help beekeepers get their honey into and get it approved into U.S. Markets, because this resin can actually remove the drugs in the honey and extract that out. And then people also use foreign sugar analysis. So this is the isotope analysis as an example. On this image, you see a C3 plant and a C4 plant. So depending on where your isotope analysis lies, you're going to get different values. So most plants that honeybees forage from are going to be C3 plants, whereas high fructose corn syrup is going to be from a C4 plant. So if your isotope analysis indicate a C4 plant, that's going to suggest that the honey had the addition of high fructose. So one of the downsides of these is that these are great methods, but one of the downsides is it only looks at single or very specific parameters. And here comes NMR, nuclear magnetic resonance. So this will combine targeted with untargeted detection of various parameters in a single analysis. So this is a very non-target approach. And what this does is it uses an external magnetic field to think of it as like shocking the proton. So each atom will have a nuclei that will be spinning in a certain direction. And in this case, it'll be the alpha direction. And for the NMR, it's going to send this external magnetic field, shock it to cause it to spin in the beta direction. And as the atom is coming back, like kind of coming back to Earth, it can the energy that it releases can be actually used to generate a spectrum by determining the molecules that were from the spectrum. So the idea of this is similar to like an MRI, where I use a magnetic field to spin a proton to create an image. And we are collaborating with QSI, Quality Service International. This Arnie is going to be doing most of this analysis here, but here's an example of one of the spectrums. This is the dark line is the sample that they are analyzing, and then the other spectrum, the other lines are going to be their database spectrum. Okay, so they ran this honey sample and they found the mannose from that peak, and that's a red flag because it's usually not observed in floral honey. And they also found the spectrum was very low on those two bottoms, and that will tell them that the sugar signals are very low there. So the sugar spectrum does not necessarily match up with, of that honey does not match up with their database. So some of the major parameters that NMR is testing, is going to be the carbon isotope analysis. So we talked about that as one of our parameters a few slides ago. So the presence of C4 sugars will suggest that high fructose corn syrup addition, for example. Water content of the honey, make sure it's around the 17% range. The sugar spectrum of the honey, the pH, HMF and diastase. These are very common indicators to see if the honey's been heated. There's a minimum amount of HMF activity should be, and also diastase. Diastase is an enzyme that honeybees secrete in the honey, and if it doesn't meet this a certain amount, then red flags are going to show up. Then there are going to be also look at foreign enzymes B and G, which are going to be used for syrup. So naturally, honey will should only contain foreign enzymes A. So here is a table of some of the preliminary results that we have. They send us a giant table and a list of the water content percentage, HMF activity, diastase, pH. It should be around like a 4. The acidity, what type of honey, and then the sugar spectrum analysis, fructose and glucose at the very end. That should be what's 70%, around 70%. And then they have like results based on that. So whether or not like the diastase was positive or negative. If they found a positive hit, that means that it was like the sample was heated to an extent and processed that way, etc. So just to sum up the table here, we're still figuring out how to best present this information. We'll eventually do like a principal component analysis just to make sure everything, just to statistically look at it. But just to sum up what we're seeing here, we actually had 35 samples that deviated from authentic honey observed in the profile of aliphatic hydrocarbons. So that's going to be from fermentation and also from organic acids or aromatic hydrocarbons, and that's going to be from like flavoring substances. 3 of our samples were actually overheated, and that was suggested by the diastase activity. And 4 of these samples were positive for foreign enzyme activity, so that'd be the amylase B and C. And then we also have 6 samples that were detected to have C4 sugars that was detected as adulterated from the carbon isotope analysis. And this is going to be from the first 69 samples that we have analyzed so far. So NMR is an amazing tool, but it's also not perfect. We saw that that sugar spectrum that they had to run against with the honey that they're testing. And in order to get that, they're going to have to generate a database. So in order to generate this database, they're going to need a lot of samples. They're going to run a lot of honey samples that are known to be authentic and create like a giant spectrum of honey. So they need a very extensive database for that. And when they're running a sample that doesn't match up with the database, even then, it doesn't necessarily mean that the sample is adulterated. It can also mean that the beekeeper just happened to be feeding during a time when honeybees are bringing in a lot of nectar. So as of now, there is still very little way to detect the differences between sugar feeding during the nectar flow compared to actual adulteration and mixing sugar different and diluting your honey. And this entire project can be challenging because of all the different plants that are out there. We saw all different ecoregions. So they're going to need a spectrum for each individual region to encompass, to capture the diversity of plants and the different types of honey in each region. And a lot of this work should be complemented with the pollen analysis, which is what we are doing in more detail. So some future works here. We are going to complete the honey analysis, at least the pollen side of things, within the next month. And we want to split the data that we have by region. So the preliminary results that I showed you, it was just for Texas as a whole. But I've been mentioning how Texas has multiple ecoregions. So we want to divide that up once we get everything, all of our data analyzed, and separate that out to see which are the dominant plants in this region. In West Texas and East Texas, because right now our data is going to be a little bit more skewed toward East Texas, because most of our samples came from there. But once we separate that out, we're going to see the major plants that we're seeing in West Texas or South Texas. So far we've analyzed about 111 samples. And for this entire study, there's only about 8 samples left. So we are almost done with that, and we're going to start getting into the detailed analysis of all the data and breaking all that down. And then we have our willingness to pay and marketing webinar on next Thursday. So that's September 27th at 10 a.m. Central Time. So make sure you check that out, because this was just one part of this entire grant. To help better market honey for Texas beekeepers. That's going to be a very awesome webinar, so make sure you check that out. In November, we'll also be presenting a poster and disseminating our results at the Entomological Society of America National Meeting. So this is going to be, we're going to be sharing our results to other entomologists and disseminating them. So this is going to be a different audience. So we're trying to make sure this research is as publicly available as possible. And one last thing that I want to talk about is the possible use of Maxent. This wasn't part of the grant, but I think that this is something that will be of interest to a lot of beekeepers in East Texas and Louisiana. And this is going to be ecological niche modeling, using ecological niche modeling with a program called Maxent. And this is going to be based on the data that we already have. And we can ask the question, like how likely is that honeybees are visiting a particular plant. So right now, we saw Chinese tallow pop up in our major plants that honeybees are, major pollen types in the honey that we're analyzing. Right now, the USDA are in talks with releasing fleet beetle, which is a biocontrol agent, to control this Chinese tallow. But there's a lot of debate whether or not this is a good or bad thing, because beekeepers and honeybees in this region are very dependent on Chinese tallow as a nectar source and to produce honey for the beekeepers in that region. So a question that we can ask using this tool would be, how important is Chinese tallow to honeybees in this region by pretty much overlaying this data and using several statistical models to make predictions that way. So that's something that might be pretty cool to look into in the future, but we'll see how time goes. With that, I think that we have some time for questions. I'm not sure how I can take them here, though, other than if the audience here has questions. But if you do have questions, you can definitely email me. That's one way to reach out.
That was great information. Thank you very much. There is also a questions section. And so if you want to send a question, make sure you can do it by text. And then we have one question already from somebody in the audience. And the question is whether this webinar is being recorded. And yes, it is being recorded. So later on, Dr. Rangel is probably going to work on getting a link so that other people can also have access to it. So again, if you have any other questions, make sure that you send those through the question information section. Do we have any questions from people in the heat area that are present?
No.
Well, thank you very much for your attention. You guys have a great rest of your day, and thank you for attending the webinar. We look forward to having you next time for the follow-up webinar for honey marketing and willingness to pay studies. Dr. Rangel, do you want to say something before we end up the webinar?
I just want to thank Pierre for a great presentation, and I want to thank all of those who attended either live or online, and we hope that this information will be useful for people who are interested in learning how the Real Texas Honey Program works and what kind of research we're undertaking. So thank you for your participation, and we'll see you next week. What do I do? Stop recording?